please.
Thank you so much. So before I start my lecture, I should say that this webinar is so informative, a lot of so good talks, bringing the scientific and the philosophical ideas, the knowledges together in this platform, unified platform, and it's a great way to start our out in look of all these matters in more positive way in future through scientific research and understanding of how consciousness works, both from the philosophical module, from scientific branches like including neuroscience, as well as the physics, mathematics, and so on. So before I begin my talk, I should say that I will speak about the neurophysiological components of consciousness with an outline of the brain areas involved. And in order to measure the neural correlates of consciousness, what are the latest neurophysiological and neuroscientific tools and technologies that we are currently having? So let's begin this with this first slide and the picture. And let us imagine a scene where two persons are looking at an overcast early morning sea. The sea is unruly, bringing up feisty high waves, and the lonely fisherman with his fishing net and a small wooden boat is trying to move against the waves to catch fish in the deep sea. So when we ask to the two gentlemen that what do they see, one gentleman says he can see dark clouds in the sky, a murky sea frothing big waves, and the fisherman with his boat. Another gentleman, he interprets the scene as he says that he see a bread-earning struggle of a thin-bodied, strong-armed fisherman trying his every learned skill to overcome the obstacles of the waves. Interesting, isn't it? We see two different interpretations from two gentlemen standing at the same place at same time and looking at the same scene. The first person's observation was very general. He spoke about all the objects he visualized, he perceived in the scene. The second person's interpreted things little differently. He was more precise, more focused on the fisherman and his skills he was using to navigate the boat on the rough sea. He thoughtfully, moreover, illustrated the scene with a metaphor where he chose the fisherman as an epitome of struggle in this tumultuous world. Though both responses show their awareness of the external situation, yet from their different reporting pattern, they represent different states of awareness at that point. One observation was totalistic, or maybe we can say global, and the other one was focal, very thoughtful. So the questions now becomes, what makes this interpretation of the same reality different? How does differential interpretation of a presented reality develop in us? Where is this perceived reality manifested? Why do we humans model the reality the way we prefer than what it is actually presented? So all these questions lead to the one area, the area of consciousness. Consciousness is no more a philosophical identity only, because scientists are now elucidating consciousness scientifically. So how consciousness builds, how it works, and changes us are the most intriguing questions in the modern science today. How does the brain model reality? So from neuroscientific view, consciousness is the awareness of the self with respect to the environment. And in the earlier days, in the early half of 19th century, orthodox neuroscientists had demarcated the awareness related to the responses only to the stimuli. But with the development of tools and technologies, neuroscience can now look beyond it. So in order to understand consciousness, we have to understand our brain first. What are the areas of the brain which are concerned? But first, we should look and understand these two terminologies. One is qualia and another is valence. So what does qualia mean? Qualia is our subjective experience. That is when we question that what is it, what sort of it, what kind of it. This can vary from subject to subject. like the earlier discussed case of two gentlemen on a seashore. Here they had two mutually different experiences from the same scene at same time. Even a qualia varies within a subject. Like a sportsman suddenly got a painful injury during a match. The attending medic asked him how he feels. Does he want to continue anymore? He says, okay, he's fine, he's okay. He has just some sort of twist and he can continue and so keeps on playing till the game is over. But after the match is over, he starts to perceive the same pain differently or rather more acutely. So here we can see that in two different environments, the subjective experience of an individual is varying. So we can see how subjective experience vary in an individual, and this is one of the factors which makes sometimes qualia or the subjective experience. Yeah.
One question. Are you sharing your slides right now?
Yeah, I'm sharing.
They're not visible. Could you maybe do it once again?
Yeah, sure. Oh, I'm okay.
Turn into host. He doesn't see? Does he not see? Yes. No, but does he not see himself?
Yes, now it's visible. Thanks.
Something, a technical nag again. I'm sorry. Shall I move from?
Yes, you can go ahead.
Sure. Yeah. So this was a picture I was saying, and I was telling you to just find it out and get a metaphor of the situations about these two fishermen. And these are the questions. We are not over. We have just started about this. So fine. Okay. Tell me one second. You can get this, right? Right? You can hear me. You can see the slides. Okay. Hello.
Yes, sir. We can see.
That's fine. That's fine. I continue. Yeah. So. There is a problem. So I was here. Or the qualia. So I was saying something about these two terminologies. First, before I go deep into this matter, the neuroscientific perspective, these are the two terminologies we should get accustomed. First, that is qualia and valence. So what is qualia? Qualia claims, as I said, our subjective experience, that what is it, what kind of experience it is, or what sort of it. So as we see that this subjective experience vary from one individual to another. Like I discussed earlier about these two fishermen, two gentlemen who were interpreting the story of the fishermen. So here they had two mutual different experience. Then I said that even this quality of subjective experience vary in the subject, giving that example of the sportsman who got injured in the game, and at that time of the game when he was playing, he didn't realize that pain so much, so he continued, and later on when he came back, then he feels the pain differently. So this is how the subjective experience can even vary in an individual. So this makes us sometimes very hard to interpret in terms of neuroscience. But I'll definitely come into this matter later on, because we cannot say it's hard, because neuroscientists, besides neuroscientists, physicists and mathematicians, they are also trying to elucidate that there must be some higher mathematical dimensions about this subjective experience. So neuroscientists are trying their best to find the neural correlates of consciousness, although some argue that how much is possible. We will come into this later on, and rather look at what is possible right now. So now we go into this term called valence or feelings. Valence or emotional experience claims our objective conscious experience, which we can measure, what one feels about an experience, that is, the pleasantness or unpleasantness of a situation. Is that thing good or bad? The simplest way to start using it is the questionnaire studies, where a subject is presented with sets of questions to respond. Here subject states his objective or emotional experience, even he can rate his experience. Psychological studies or psychological experiments are another useful choice to do this. We can record subject's reaction time to a presented stimulus and understand his actual feeling or the objective experience from it. besides his verbally written or a verbally reported response. The idea is that in some cases, if a subject does not wish to reveal his actual feeling inside about that stimulus, it cannot be hidden from his reaction time to that stimulus, as reaction time between spontaneous and thoughtful responses vary. So this can be also helped with different physiological monitoring devices like heart rate monitoring as well as the electromyography in order to get the physiological responses associated with different experiences and feelings. Here I want to say about, okay, we get the feelings, but can we locate these valence or the objective experiences in the brain areas? We do. We have some techniques. I will move on one by one, but right now I will put this slide and show you that one of the most fascinating one is that we can look into the brain of us through this task. When a subject is performing a cognitive task, or rather when he's going through a sensual or sensory perceptual experience. So these areas of the specific areas which are related to that particular function, so cognitive task or the sensory experiences, will show activations, which here you can see through this green blob. So through functional magnetic resonance imaging techniques and other imaging techniques like NIRS, SPECT, we can look over the brain, and if we can find out how these different brain regions at what time point they are already they're all together coordinating in a different task. Now, an important question comes that if our responses mean all about consciousness, so does that mean that a subject who is unable to respond, give his expression, what do we say to them? When they are not reacting? Are they not conscious? Are they unconscious? The answer to this question is very important to us to know about the coma patients who are unresponsive and are in life support systems in intensive care units of hospitals. So a recent study on acute brain injuries in patients has shown otherwise. In these patients, traumatic brain injuries and hemorrhage caused cognitive-motor dissociation task, a condition where there is a detachment of sensory and motor coordination. Behavioral category enrollment of these patients, they show they were in coma. Some shows unresponsive wakefulness syndrome, and some minimal conscious state in their immediate early days of injury, because just after the injuries, they were brought to the hospital. And then these category enrollment the clinicians did on these patients. Now they did an experiment. They record brain EEG and then gave a task while they were recording the brain EEG of these this coma patients. This gave two tasks. One, you can see here, the command, the verbally given command is keep opening and closing your right hand, right or left hand. And the orange one is depicting that you stop opening and closing your right hand. So these two commands are not just only the auditory stimulus that will go and get sensed in the primary visual auditory areas of our brain. It should be interpreted properly, that there are two mutually opposite things in the command. One to open and close your right hand, another what the task you are doing, you stop doing these tasks. So these commands were played and the EEG were recorded. And after the commands were played and the EEG were recorded, so they took this EEG and they put it into a machine learning algorithm. And then what they see is that from their brain EEG activities, that these coma patients, they can actually understand or get the commands, the meaning of they could listen and interpret the meaning of the command. So here you can see in this orange, these graphs, these are the decoding prediction from the general individuals, the normal people like us. And from these decoding principles, they use this machine learning algorithm and predicted what is happening in the patients. And you can see the patients in the blue who are having this, some of these patients, they can follow the task. they can show the same level of this, the same structure of the responsiveness, although the amplitude varies different. So this shows that they conclude that these clinically unresponsive patients, they have brain activations at two mutually opposite motor commands. This is important in a sense that as the commands are meaningfully different, it needs awareness to understand. And here you can see on the right side of the screen, the spatial EEG pattern of the healthy volunteers and the cognitive-motor dissociation patients. And we can see that the visual area, which is behind, and in the left-hand side of the temporal cortex, the CMD patients, as well as in the healthy volunteers, they show some kind of activities in the alpha and the beta range of the EEG. Now, at this point, let us look at our brain. The anatomy of the brain shows that it's a highly convoluted mass of tissue outside, called the cerebral cortex. This mass is the center of several functions, such as perception, memory, cognitive executive functions, thought, creativity, intellectual abilities. Deep inside and caudal to the cortex are two diencephalic structures. Just here, the thalamus, which we commonly know as the relay station of sensory informations, but nowadays we know also that it participates in central perception. The other diencephalic structure is the hypothalamus. Below it, it is a key regulator of cardiovascular and endocrine functions. Further caudal to the diencephalon lies three structures in rostrocaudal direction: midbrain, pons, and medulla. They are collectively called the brainstems. They modulate our state of vigilance, arousal, sensory and pain perception, voluntary and autonomic functions through the ascending connections to the diversified cerebral cortical areas, thalamus, hypothalamus, and through connections ascending towards the spinal cord. Behind the brainstem, there is another structure called cerebellum, important for modulating our voluntary movement. Cerebral cortex holds the center stage of neuronal computations of our brain, which extracts complex information from our outer world. From outside, cerebral cortex is divided into 4 lobes, and these lobes can be easily detectable based on their position with respect to sulci, the crests in the cerebral folding. Areas of sensory perception such as vision, hearing, touch, proprioceptions are spread across the parieto-temporal occipital lobes. The parietal lobes lies behind the central sulcus, and behind parietal lobe is the occipital lobe. The temporal lobe is below the lateral sulcus. Deep inside the temporal lobes lies the amygdaloid hippocampal complex, more precisely the limbic system, where our explicit memory, emotion, and feelings are processed. That is the qualia and valence part. The frontal lobe, before the central sulcus, is dedicated to motor programming and execution, attention, cognitive function, decision making. Thus, the frontoparieto-temporal connection builds up our subjective experience at that point. So we now know. So this is the qualia part. Now, look at the general principles of sensory perceptions. For example, taking here the visual cortex or the visual perceptions. The first rule of the sensory perception here is information processing takes place in a sensory pathway, and which is actually hierarchical in nature. How? For example, when the information of a visual scene falls in the retina first of our eye, and then passes serially from down to upstream areas in the visual pathway, upstream visual areas abstract more complex information about the scene than the downstream areas, like contrast, luminance, spatial, temporal frequency are the most downstream areas in thalamus, lateral geniculate nucleus. Then in the intermediate area, that is striate cortex, we call it primary visual cortex. It interprets the informations of the color further into orientation as well as the depth perception. And then these informations are further channelized into the higher brain areas called the extra striate cortex. And these extra striate cortex or higher visual areas, they decipher the informations, more complicated informations that about the subject, how is moving, its motion, its perspective, and what the subject is in complicated details, like what is the object, even the face recognition task. So this goes on in the much higher extra stride areas of the visual cortex. The second general principle of sensory perception is that sensory perceptions are processed in parallel pathways. This is another interesting thing. Besides, the information is going serially as we move into the higher levels of our structures. These informations, they are certainly divided out. That which information, a particular kind of information is perceived in one kind of pathway, another. So what are these informations? One is the what pathway, another is the where pathway. Actually, what is it, what the subject is, how it looks like. These are the informations. Just the details of the subjective analysis is done in this case, in this visual pathway, in the ventral pathway, which is the inferior temporal lobe. You can see from the primary visual area, which is V1, information is passing downwards just below the lateral sulcus, superior temporal sulcus. This is actually a monkey brain I'm showing. And then go to the V4 areas, then into the higher ventral temporal areas. And there is another information that is where means the location of the object, of the subjective thing that you are experiencing in the space. So this is done by the dorsal pathway. The dorsal pathway is the parietal pathway, where the perspective, the motion of the subject with respect to this to the background, these have been analyzed. The third principle is that the sensory informations of different sensory systems. So like visual sensory system, we have auditory sensory system, the sensory systems that is process the sound. Then we have the somatosensory system, which processes the touch, the proprioceptions, that the position of our hands, legs in the space. And similarly, we have the sense of our taste, our olfactions. So the generalized thing is all these complex informations first about the location and the type of the context. These have been processed separately at the higher structures, and from these different sensory systems, they need to be integrated. There is a purpose. There's a purpose in order to understand the information from the external world meaningfully. So all these areas, sensory areas, they need to integrate in one area. So this is the called the parieto-temporal convergence, where all the sensory information from the same different sensory modalities actually gathered. Now comes the important questions, like if you see, you get this idea now that how subjective experience are responsible, but we need to see here one thing, that how is the unified percept it formed? Means this is one of the intriguing questions, like the what and where information stream, how is it integrated together in the sensory modalities, across the sensory modalities? So this is actually called the binding problem in neuroscience. So what does it mean? The answer can give us the knowledge of how subjective experience of qualia works if we can solve the binding problems. One of the postulations is that we use our attentional abilities to integrate the parallel information in the scene. This can be done, or can be one of the this can be one of the reasons that why interpretations of the same scene, if you think, just before I explained about these two gentlemen, varied. The factor could be their attentional selectivity behind their differential interpretation of that scene. So selective attention is the component for binding the parallel information of sensory perceptions. So what does the attention do? It maintains our internal state of mind. It helps to bind this complicated information from all those different kinds of the sensory pathways together and gives a meaningful information about the scene that is being presented, about the reality. So it maintains our internal state of the mind through it, modulates awareness, its priorities, expectations, and operates from different levels of the brain, like in the frontal lobe and the superior colliculus. Now, if we see on our brain and then neural activities, I will come later on, you can see that these ongoing neural activities are correlated with high frequency postsynaptic transmissions, those areas, that is the neurons which are responsible for attention, used to bind these whole things, which might be one of the modulators of behavior in context-dependent manner. Now there are two important questions, the two other cornerstones behind our consciousness. Our perceptual experiences are necessary for our development, we know, and it takes ages. So how was such countless perceptual experience integrated to our consciousness? One through learning and another through memory. So what does learning do? So we learn to know what to attain, what not to attain, how to master motor and cognitive skills, how to explore our world around us. We consolidate what we learn in form of implicit memories and explicit memories. First of all, I will go through, I will just explain what the memory first here. So what is implicit and what is explicit memory? Implicit memory is the memory which doesn't require our conscious recall. For example, if you are driving a cycle or opening the cork of a bottle, water bottle, you need not always need your consciousness in order to do this kind of tasks. This you have already mastered. So this is already inherent in your brain, this kind of memory. They are non-declarative. You can do it. You need not have to remember what to do next, paddle this, go on paddling on this side, then this side, when you are an experienced bicycler. So these are kind of examples of the implicit memory. So that's why it is required for the perceptual and motor skill. So the other kind of memory is the explicit memory. What is explicit memory? Explicit memory is the declarative memory. It requires our conscious recall. When we actually speak of an object, or when we think of places or people, we have to bring all those things, the situations where I meet the people, at what moment of time, this whole information sometimes comes together in our brain. So this memory is called the explicit memory. So we call it a declarative memory. And declarative memory, how this encoding and retrieval goes on. This is actually important to understand, because a lot of things also depend upon this memory and learning. Hippocampus, we know, is involved in spatial navigation and episodic memory. Spatial navigation, we know that when we are navigating in a place, for the first time, we have to go through the landmarks, the important landmarks, and keep on that in our brain and understand about the place. And next time when you are there, you will realize that you are no more aware of that situations that you were first in going into that place. So somehow these kind of informations are integrated in your memory during the navigation process. And episodic memory means when you are bringing some memory back from the time, from the sequences. So what are these areas? These areas that are that encodes and retrieve memory. So the multimodal association areas, we know, we have just gone through this sensory perception scenario, and we know that from the sensory perceptions which are taking place in the higher order multimodal areas, where the things are integrated by the attention, they goes to the entorhinal cortex. From there it passes to the perirhinal, transmits into the hippocampus, where their consolidation goes on for the first and the retrieval also takes place. And then to the subiculum, it is another area, and it takes its exits into entorhinal cortex, perirhinal cortex, and again to the multimodal association area. So this is the route that we use up our sensory experiences and try to consolidate the experience which we think through our learning is attainable, or we can throw it out. But this consolidation about the declarative memory goes on in hippocampus. So how does it go on? Now hippocampus forms memory engrams. What are these engrams? These engrams are actually structurally a group of cells whose activities are actually synchronized through strong biophysical and biochemical changes. Let's see how it is done. So engrams, they form a stable map of a memory trace. You imagine this scene. This is a space, and the person in color, if you can see, the green, red, and the blue one there in the distance, besides the small dog, it is you at that different, in the entire scene at different points. So you are navigating this environment, right? And this is this cluster of the cells. So when you are here, looking at this circular sculpture at that particular scene, one of the cells in that engram, it fires. It starts to get that firing. And whenever you will come into this particular position, you will see the cell is firing there. And then when you move to another position, like here, going to a vendor asking for an ice cream with your friend, and then you will see the green one there. There is another cluster which is encoding that environment, that spatial part. You move backward, the scene from where you entered, where you met your friend for the first time. You see there the spatial information, the time informations, they are being also encoded. Now you move forward with time, you left that places, everything goes on, and you come back after several years, seeing the scene from a different perspective. What you will see, you will recollect every event or whatever that has happened at that time when you first visited at that place with your friend. You met your friend, you went to that sculpture, you took an ice cream from the vendor, whatever it is. So how is it possible? This is possible because the entire thing is engrammed in your brain. So this is how the engrams works in case of our declarative memory. One thing you can see that through these engrams, you can move across time. That is, you can move wherever you can. You can remember your childhood, or you can every points of times where you have collected the information. So you do not have to enter that scene from a specific point, like from behind. You can take the scene from any perspective, from any dimension, and you can interpret the scene there. So learning is the basis of the biological individuality. Habituation, sensitization, conditioning, overt learning, they occur at several levels of the brain. Because it is important to learn before we want to memorize. And the molecular to anatomical manifestations of learning spans from gene expression, protein synthesis, long-term potentiation to synaptic pruning. Learning alters the anatomical organization, a phenomenon, we call it plasticity. And what happens through learning? That when we started playing a guitar or some instruments, or even we started to play cricket or kicking a ball, that these connections through learning of the afferent pathways, sometimes the connections which are needed to acquire a particular skill, they expand, and that what is not needed at that point, they retract depending on that activity. So you can see how we can modulate our own brain through our own will. If we learn something which are positive, it is definitely giving you more positive impetus to your attitude, to your personality, to your brain development. And it takes place at any moment of time, because the developmental things, it happens, the circuits and every structures at the earlier age, there is a critical period, but that is set. But what kind of tract or what kind of areas you want to grow, that depends on you, and you can grow in all ages through your learning, through recitations, through music, through creativity like art, through playing, everything. So it has a bigger role in our life. So at this point, what we understand is that we get to know about this sensory perception, the sensory pathways, how it works, about the anatomical organization of the brain, about the attention, an outline of memory, an outline about learning. Now we want to look that we have to integrate all these things, the informations together. When a person is experiencing a subjective experience, how these different physiological components, we are saying they are the correlates of our consciousness in terms of neuroscience, they are doing, they are interacting with each other at a particular subjective experience, and how they're varying from one time to another time. This is actually very, very difficult at this moment of time, but there are technologies. Definitely what we can do is that the first thing we have to understand that we have to look at the level of the neuronal wiring, neuronal communication, functionality at each level of this brain, of the brain, like the level where the attentional process are modulating, the place of the brains where the memory is consolidating, the entire part of the learning where it is taking place, and the sensory perceptions. We have to look at each level of the brain, and we have to look between the cortico-cortical connections at the level of the cortico-diencephalonic brainstem structures, between the hippocampus, parieto-temporal, prefrontal axis. at the level of the laminar to the columnar to the microcircuits. You have to go to the synapse and to the genetic level at the same spatiotemporal window in order to understand the whole thing. So neuroscientists are mixing various kinds of techniques and technologies in order to understanding the firing of a single neuron or a group of neurons related to postsynaptic potentials at microcircuit level to electrocortical graphical activities over larger cortical areas in behaving animals. So these techniques are not all applicable in humans. There is no possibility, right? But because some of these techniques are, most of these techniques are invasive. You have to just put the electrode inside the brain, which is mostly not the case in case of our human studies. As well as some techniques also I will show you, will use the genetic manipulations. But here we integrate the animal and human behavior together in order to understand that there is always an evolution, a basic level of understanding of how the neurons work. So at what level we can go to understand this whole scenario. Like we can catch a cell and find its firing rate to a particular behavior, or even we can catch a group of cells at the different levels of that point and of that particular area and see how they are responding to a particular subjective experience. We can get the local field potentials, that is the postsynaptic activities in that area, the electrical activities which are going around a few hundreds of populations of neuron at that particular point, and how they are correlated with this individual firing of the neurons. And in the greater scale, we can also do the electrocorticography. That is, over larger areas of the brain, we can find the synchronizations of the correlation of the activities of the other areas of the brain, and we can correlate these all things, starting from the single unit firing to the local field potential, with how at the time point and in the space, the whole thing is going on in any kind of subjective experience. So let's see, one is this, is the somatostatin expressing interneuron in dentate gyrus. I'm putting one of my work here and unpublished data, where you can see this is the dentate gyrus in hippocampus, which is important for memory retrieval. That after a long time, when you are going to some place, or not that place actually, particular place, a place which is almost similar to something, an experience, then this kind of this area, it actually helps you to retrieve this information and to match the similar experiences that you have earlier experienced in your life.
You have 3 minutes more.
How much?
3 minutes.
Hello, Mark. How many minutes left?
You have 3 minutes more.
Oh, okay. Okay, so this is the way we can go through by understanding that. Okay, I finish it. So we can understand through these neuronal activities, and from the LFP and these integrations, we can get how the microcircuits work. There are other smartest techniques like the neurotagging. So when an animal is performing a behavior, when you put an animal in a fear conditioning situations, you can tag the cell genetically, and you can later see the populations of the cells which are actually responsible for this fear response. And one of the smartest techniques now, it's actually there in the neuroscience, is optogenetic manipulation. So you select this particular population of cells, and you can genetically insert a light activating protein and then you can into that particular population of neurons, and you can just modulate this, means switch on or switch off the particular behavior by switching on and switching off this population of neurons by shining light from outside. So this is an example you can do, that when you put a mouse inside an environment where naturally fear is evoked, so the cells which are responsible for evoking this fear, you can genetically or you can optogenetically manipulate to repress this cell, and you can see that his fear is gone. He is just behaving what he has behaved in the neutral situation. So these are techniques, as I said, they are not being used in these in animals, but we have techniques like already been discussed earlier, like EEG, yeah, definitely EEG and fMRI. So here I can show you what EEG does. It differentiates a brain between a working brain, a thinking brain, relaxing brain, and in a wake and a non-wake in a person. So you can see here how it looks like. Here you can get these small amplitude waves. As we know, There's low amplitude waves, the alpha waves by now, which are between 8 to 13 hertz. You can see it is a recording in an individual whose eyes were closed, and this is a recording from a single electrode placed on the prefrontal lobe, on the left prefrontal lobe, that is on the over the forehead, I will say. And now this is the task. What is the task? The task is that the subject is the oddball paradigm, has to look at the screen and has to respond by pressing a button in the computer whenever he sees an odd stimulation, odd orientation. Like the opposite orientation, it will be the horizontal orientation, which is presented in a very low frequency, random frequency. So this shows that the task changes the spatial structure of the brain activity. The alpha waves here we can see is decreased. So this can show that the thinking brain, how it looks like. Next, I move into another aspect, that sometimes our neurons, they represent our reality differently, like the illusions. Okay, let's move out from here because I have a shortage of time. So now I move into the last part, is that can neuroscience, it elucidate the cultural and traditional influence of consciousness? This is a bigger question. So for now, we summarize, we create an internal representation of the external environment through what we sense, how we perceive and learn and store in memories. We build thoughts and feelings and express them in our action, in language, and through creativity. Thus, our brain exhibits the neural representations of our conscious behavior. When we speak of human consciousness, we need to acknowledge bigger influence of human culture and tradition, a prime external factor behind its development. Habits, etiquettes, morals, intellect, thoughts, logical thinking, they are influenced by culture and tradition. This opens up a new venture to study the impact of ancient Indian Vedic Upanishadic teachings in the modern era of human consciousness. The importance to investigate these from the neuroscientific perspective is manifold. What else can be better than to understand from the perspective of neuroscience Swami Vivekananda's scientific and philosophical ideas of yoga, mind, and consciousness based on Vedas and Upanishads? Although the task is not trivial at all, still neuroscience offers immense promise to decode neural correlates of consciousness along with interdisciplinary approach from areas of science and philosophy. If we turn back the pages of the history for a while, we can see how some of the India's greatest sons were deeply influenced, inspired by the thoughts and philosophical ideas of Swami Vivekananda. Their acknowledgements, along with their greatest deeds, are the proof of their supreme consciousness. Their lives and the practices are the glittering examples of how we can positively influence our living. Thank you so much.